Let's begin with the prediction. So the meaning of sequence variance. So first of all, again, let me repeat, evolution selects a neutrality of proteins against changes, or such that the ones that we see are copies of things that somehow where change is neutral, right? But with respect to sequence variation, what I now mean is not necessarily in the sense. It's not necessarily in the evolutionary sense. When I ask, can I predict the effect of a sequence variant, then I'm talking about a chance sequence variant. How can these variants happen? What type of variants are there? So let's go onto the level of the genome. On the genome level, we have substitution, so that's the typical regular, the simplest change. You have one particular nucleotide that is changed against another nucleotide, okay? Then there are simple insertions and deletions, so you take something out, you add some genetic material is copied, so you can copy it at some other position. This is a sort of a particular case of that, the duplication, where you duplicate an entire region somewhere else. It can be a repeat directly thereafter, so region, and can be a repetition somewhere else, a duplication to somewhere else. There's an inversion. So there's a way in which you read the DNA sequence. Obviously, it's a 4-letter alphabet, and the 4-letter alphabet read in that direction makes different sense than read in that direction, right? So every 3-mer is coding for an amino acid, or at least of the coding part. And again, it's not the same word in both directions, just like words are not. There are these exceptions, but many words are not. Now, this does in fact happen that you essentially, you cut a piece out and you insert it the other way around. So that's an inversion. Again, those are larger changes. What we will mostly be concerned with is these small ones here. Now, on the level of proteins, we have substitutions again that are the single amino acid or the single nucleotide change can lead to silent or nonsense and missense variants. So missense means that the nucleotide change leads to a change in the amino acid sequence. In the other cases, essentially doesn't. And just like on the level of the genome, the insertions, deletions on the level of the genome can be translated to the protein. There again, you will have insertions and deletions. And in fact, inversions can lead to different amino acid sequences, because the word the other way around means something else. Now, here are a few more words. The most important one here is what I'm going to use mostly, a single amino acid variance. I will briefly talk about something that I call 19 non-native. So when you mutate at a particular amino acid, the native is 1, and then you have 19 alternatives to the native amino acid you have, right? That's what I mean by 19 non-native. So essentially try everything at that particular amino acid that is not native, that is not in nature. Now, SIFT possible, so I mean, those are nucleotide changes where a single nucleotide change, I'm sorry for the slide here, for the overlap between these two pictures. So let's look at the nucleotide UG that happens to code for an expensive boot, but also for a tryptophan. And when you have these other two here, both of the UUU and UUUC, they would code for an F, but you see this is not a single nucleotide change, this change, right? So you change an amino acid through changing 2 nucleotides in order to get to an F. So in that sense, it's not SIFT possible. You cannot get to that different letter here. You cannot get from a W to an F with 1 nucleotide change. Okay? Now here we have the UG. If I just change the central G to a C, I do get another letter, another amino acid. Okay, so that is SIFT possible. So here, 1 nucleotide change leads to a change in the amino acid. Here, not. Not a single one, at least. A double one does. And this sort of goes back to the code of the nucleotides to amino acid. You see which single nucleotide changes change the amino acid and which don't. Again, I briefly talked about that. So there's the sickle cell anemia the sickle cell disease, where a particular a single amino acid change leads to a disease form. Okay, now let's talk about first methods. And again, the background problem of developing methods that predict the effect is really that we have to sort of fight against the neutrality that we observe in evolution. And that brings us to the same story that we talked about briefly last week, which is the simple idea of using alignment information or using the blossom matrix. The method is called SIFT, for sorting intolerant from tolerant. And the idea is, as we discussed, you simply essentially ask, what is this particular change in amino acid doing to the biophysical potentials? Or put it differently, more specifically, look at the blossom matrix that reflects what residues you can in nature change against each other without sort of affecting the protein function. Take that matrix and see whether that particular change is compatible with that matrix, or whether that matrix would say that that change, that variant, is in fact a neutral variant. That's in principle the idea. PolyPhen, historically called polymorphism phenotyping. Ultimately, polymorphism is in fact a single nucleotide change, a single amino acid variant. And now here the idea of PolyPhen is you're not looking at an average feature difference, but you're looking at a family. And you're asking, in that particular family, is the residue that is conserved the one that mutates? Or is the variation that I observe, the particular, in this particular case, tryptophan to S that I had there, is that one that you see at that particular column in the alignment? If yes, then you say this is a neutral change. If you never see it, you say it's a disruptive change. In particular, it's very disruptive if the column is entirely conserved, so everybody in the family has an E there, and you suddenly see a V. Then you say this is a very disruptive change. Okay, that's the idea behind it. Now, here's SnapS3D. The idea behind that one is, well, do the same as before, but now, whether you're on the outside of the protein or in the inside, it is something else. So, yes, you look at the conservation in the alignment, but the conservation in the alignment means something else on the surface and in the inside. In the surface, the conservation, there's a lot on the surface that can vary, and that's not really important. And the few that are conserved are important sites. In the inside, rearrangement will lead to misfolding. Misfolding will lead to a functional impact, although it's not really affecting the binding site, but the protein doesn't fold anymore, therefore it cannot function. That's the idea behind that one. And those essentially you may call the first sort of generation methods. They do not really use any data fitting. They just use an idea what you could do. You need a few samples to just see does it work at all. But essentially that's the idea. You need to fix a few thresholds. That's it, right? And then you can use every data that you get to see how well your methods work. Now let's get back to the data. On the level of the data, I'm talking about the effect upon function of sequence variance. Now there are different effects. There's the effect on the level of the organism. So these two mice here look different. In this particular case, again, this difference is caused by a single change. In this particular case, it's a single gene. There's not a single, but I believe you can induce something like that. At least you can certainly induce hairlessness, naked mice, by a single amino acid change. As opposed to situations here on the other extreme, where you have detailed biochemical or chemical measurements that measure the energy or the delta G, the free energy of binding, and they see whether that particular variant affects the binding of these particular two mutants, or these particular two proteins, okay? It could be on the level of the structure. We sort of see that they change, and it could be something that we just see as a change in color. So the medium is different, and there's somebody looking at the color and says, well, here's change, here's not change, okay? And these are very different effects on very different scales. And when we put them all together, then we get a data set that is roughly 40,000 effect and roughly 14,000 neutral. So a quarter is neutral, 3/4 are effect. Now, we do not know. It could be that this number that I see here is reflecting what we will see in reality. So if I mutate in a particular protein, so people have done that experiment, and even that one, so we mutate every single residue in an entire protein, and even that is not a complete answer to the question. So it looks as if a lot of the variants are neutral, and a lot have some effect. Now, we do not know whether that ratio is right. What we do know is what we have in the experimental data set is biased toward not having enough neutral. Okay, we know that because people don't publish something that is neutral. Okay, that's another extreme way of putting the story. Some experimentalists say no variant is neutral. Every variant has a little bit of an effect. The difference is really the scale. So some have a dramatic effect and some have a tiny effect. So this entire concept of neutral, they argue, is misleading. And there's some truth to that statement, okay? I'll get back to that at some other point in the future. In terms of machine learning, and that is now we are beyond having to fit a single parameter. Now we are in a realm where the data set may be small, but this is big enough to really think machine learning. This, in fact, we sort of collected this data. This data set here to do machine learning. And if you do machine learning, so in some sense the major problem, other than that is small here, is the imbalance. We know there are ways around imbalance. What are they? So if I have a very imbalanced data set, what can I do?
Oversample.
Oversample. That's one way. What else can you do?
Undersample.
What else can you do? So you can change the way you essentially rate each particular sample. So you can weight it. And then there are algorithms that do it, which is sort of something like weighting. Smooth is one of those. So part of packages. But ultimately, these are the kinds of things you can do. Now, another thing that you could do is try to see whether you can somehow get more neutrals. And the way we did that here was we followed the following idea. We argued if you had two proteins here, this one, and these two proteins are both experimentally characterized, and for both the experimental characterization says they are enzymes, and they have a particular enzymatic activity. They're hydrolase with these three enzyme, or with this entire enzyme classification. Both of them. Okay? Now, if these two are very distant, so I have 30% sequence identity or something in that ballpark, I do not know whether the changes in between may matter for something else, because maybe they are enzymes, so they have an enzymatic activity, but maybe they do something else. Maybe one of them binds DNA and the other one doesn't. And that happens in the residues that are not conserved between the two. If, however, these two are very similar to each other, say they are 80% sequence identical, so they don't have entire regions that could possibly bind DNA that are different. They have a few residues in between, and we know they have the same function. Then I can argue that anything in between, or anything where they vary, is in fact neutral. Because I can observe the function. What is in between is not big enough to really adopt a new function, so most likely I argue they in fact are neutral variants. And then I can use that as a data set to enrich my neutrals, and this in some sense is sort of a simulated data set. And SMOOT is one of these functions that tries to sort of simulate data to not oversample but to equal sample. So you could essentially dig into your data set. So that's another way of doing it. That's the idea behind SMOOT. Let's get back to that point. You could dig into the negatives and say, and ask yourself, are there others that look very similar? And since they look very similar, I can create an in silico neutral by creating things that input vectors that look very similar, that I assume are very similar. Essentially, like creating an artificial protein, and since you create it, in the protein world that is not easy. Sometimes for imaging, that typically is relatively easy to do and gives you good results. Anyway, we end up, in this particular way, we end up with a data set that is fairly balanced here in terms of neutrals and effect, in terms of numbers. It's not balanced in terms of potency. So we should not forget that the experimental data is here, much more on the positives, on the effect variance, than on the neutral variance. So what I mean by potency is this addition, or these added neutrals they have something in common. So they are sampled from families that are similar to each other. That's one thing. There's an overlap, a sequence similar to each other. And there is a mistake in my set. Okay, in the experimental data set, there's also a mistake. But I know that there are variants between, for instance, particular protein in human and chimp. Although they are very, very similar, there are differences in terms of the way the protein is digested by another body. I could not take the chimp protein into my body. My body would reject it. In fact, my body would even reject a donation of any, in some cases, of organs or even other proteins of other people in some cases. So, although they are very similar, there is a difference, right? And that reality sort of puts a mistake into my neutral data set. Okay, anyway, I can now begin to do the second generation method, which essentially build these larger data sets and do machine learning. I'll get back into that method. I begin with the first idea about that method now. And then there's sort of a third generation methods that try even larger data sets and put in more information. Okay, and the method, sort of our in-house method, comes originally from Jana Bromberg, was a professor at Rutgers University. Let me just go through a bunch of methods before that. So here is a method, and I'm rushing through them in a way that means you will not be asked to remember any of these. I'm just going through them to give you some ideas how they do it. So here's an idea that comes from the Blondel lab. And the idea is that when you sort of go through the thermodynamic cycle of binding, you change your state, and this change in state is induced here by mutants, and for these mutants essentially you see how much the energy changes. Okay, you have a particular mutant, you see how much the energy changes, or how much this cycle is here affected, and this is what the machine learns as data. Essentially, learn the effect of a variant upon the energy of binding. Okay? In terms of going through some thermodynamic cycle. Here, a method from Marian Ruman's lab called pop music, and pop music does this in more explicitly, and I'm going to show you three other methods that do that too. More explicitly means, in this particular case, this method looks at particular binding sites, protein-protein binding sites, and the effect upon these binding sites. So is this variant, how is this variant here affecting the energy of the binding of two proteins? That is what this method answers, and those are the data points that the method uses for machine learning. Okay? You essentially create an energy model of proteins that bind, and you see that you put a mutant in, and you ask how much does that mutant change. Okay? Now here is a similar idea, but rather than sort of computing the energy, and it's from Emilio Capriotti and Rita Casadio and Bologna, Piero Fariselli. And here the idea now is, instead of computing the energy, just take measurements of delta energy of binding of two molecules and learn those in machine learning. Again, all of these are very different data sets. For all of these data sets, and let me just keep going, I believe there are three more. Here is the one from Josef Weizmann. This is a slightly different data set of delta G, but other than that, a similar idea. Josef started with more geometrical model, but also ended up with machine learning. Here's an idea of a tool from the Sander, or Boris Reva, Chris Sander. So again, the question is which mutations are functional. In this slide, I believe I need a better slide. So the idea behind this slide is you have a particular position here where in some part of the family you have an E, and then it flips to an N. There's another one where you have a conservation of a G. Now, this may allow you, or may tell you, okay, there's something that happens here, and there's something that happens here. Okay, we know these are important sites. But beyond that, what I know is that the part of the family that is above this, so the part of the family that's the blue here, they have something in common. So they all have something in common, but they have something more in common. These top 4 and the bottom 3, right? So those 3 are more in common to each other, those are more in common to each other, and there is some switch, okay? Now this switch comes with an amino acid change. So the switch from one part of the family to another part of the family, again, everybody in the family is doing this, has sort of the same function, but this switch has an important effect, okay? So in that sense, this particular variant here is one that has a dramatic effect. So it sort of does not only estimate, do I see the N sometimes, and do I see the E sometimes, but it asks in which one, if I see the N in this one here, although the N is conserved in the family, it would say, no, that's wrong. If I saw the E in this part here, same story, that is wrong. Although, in terms of numbers, of the 7 counts I have here, 4 are E's. So having an N to an E should be absolutely accepted. By most of the family members have an E, so it should be absolutely accepted. But if you're one of these here, it shouldn't. Okay, that is what this idea behind that method. It's not that easy to automate that, but that is the idea behind this one. Here, from the same lab, so including Cassandra, but originally from Deborah Marks, Thomas Hopf, comes sort of the next step in that direction. And the next step in that direction essentially says, okay, so these particular type of variants that define subfamilies, this is something that allows me to look at subfamilies, and within subfamilies it looks at what is important, what is too important to be conserved. But it means that I have 2 positions that are importantly correlated. Importantly correlated means there's something. For instance, one reason for the correlation could be that at some point, somewhere they are close in space, okay? There's a connection between them. And that links these 2 different columns. And if I could sort of invert this problem by seeing the correlation, and then I sort of predict proximity, and I apply that to protein-protein interactions, then I can essentially not only predict which are the residues inside of a protein that bind, I can predict which proteins bind, and I actually can predict what the effect of a variant is. Because now for every single column I can compute its overlap with all other columns, and I can say that some are more overlapping or have more coupled connections, and I can simply compute the energy for that event. And that created a method, by now it's published, I should actually change the quote here, that essentially allows you not only to predict that a particular sequence variant affects something, but actually also how pairs do that. Because now you see all the couplings in a protein. It's a very complicated method. It only works if you have very large alignments, and it's one of the few methods that is not available as a tool just because of its level of complexity. But for whenever it works, it's an absolutely great tool. Now, how do we compare methods? And typically the answer, and I've talked about that before, is something such as this. So you have a bunch of method, you have a bunch of criteria, accuracy, sensitivity, specificities. Here the confusion matrix, true positives, false positives, true negatives, false negative, and so forth. And here you have actually the methods. And this is the kind of thing that you do in a thesis. This is the kind of thing that you do deposit as a table somewhere. It's not the kind of thing that you do in a talk. Not in your thesis talk. It's not the kind of thing you put into a paper as a main point, because it is very complicated. So if this story would now be such that one of them, I mean, here you begin to see in one column they are also relatively close to each other. Yes, sensitivity and specificity, so some have a very high number here and low number here, some have a lower number here and a higher number there. So there's an anti-correlation because all of this somehow is an accuracy. But the point being, when you do your master thesis, when you do your PhD thesis, you spend a lot of time with the numbers. You become the expert in your numbers, right? But communicating small differences on this level is complicated. And that is not only difficult in terms of communication, but it's actually also complicated because the longer the list, the more you find things that are similar, the more you find things that are different, and the least you will actually see something that you can say, well, the main result here is this. Other than your main result is they all have advantages and disadvantages. That could be a conclusion from something like that. So that is one problem. The problem typically it has to do with the fact we don't actually really know what the single number is that we want to measure. We do know that specificity is relevant, precision is important, recall is important, but we already, when we see some method has a high specificity, precision and a low recall, another one has sort of an average of both, a little bit of both, right? Which one is better? That depends on your goal, on your task. How do I then compare these methods? Well, people introduce the F1 score. That is sort of an average between the two. But again, this is not quite capturing the story. And ultimately, the problem always is that people will apply your method to tasks that you don't foresee. Okay, so you don't have this one score that you can measure and say, this is what it is, right? Sometimes you know that it will be used for that purpose and you can measure or estimate it under that purpose. But not very often. Let me look at something else. Let me first get into the in-house prediction method that was started by Jana Bromberg, that started with a simple neural network. In fact, we tried SVM, we tried all kinds of other tools, and they did not work better. Deep learning, we have 40,000 samples. This is absolutely not a case for deep learning at this point. We have tried again to do slightly different networks and to sort of project it onto a problem that is deep learning. That is something that the one who's doing the videos, Dimitri, is trying to do at the moment, by ultimately learning how do sequences look. And can I essentially, when I look at all the sequences, can I predict why some sequences don't look like that or something like that? That ultimately is the idea. Anyway, in this particular case, we have the story that I talked about last semester very much, and for the binding sites this semester, so I have, in this particular case here, 13 consecutive residues, a window of 30 consecutive residues, cut out from a protein. This is each amino acid here with 21 input units, and I predict effect on neutral. So all of that is very simple. Now the question is, what are the input features? Well, we will clearly use the alignment information. We will use something about the level of the chain. We will use somehow the biophysical features here, such as charge, hydrophobicity. Oops, I should get the animation for that one out. I'm sorry. Now, one important one here, and briefly we talked about it on the whiteboard, all of the input features will say something such as is not conserved, it has a charge, it's polar, it's hydrophobic, but it will also put a feature in that has to do with a change in that feature. So it's predicted in a helix, but also if I put now this particular new amino acid in there, or if I change my native against the variant, how is this prediction changing? So if I have 13 consecutive residues, is this additional residue changing the overall charge of my window? Is that changing the prediction for helix? That's another set of input features that turned out to be very important for some cases. So solvent accessibility is something that I did not talk about. Secondary structure I did. Residue flexibility I briefly did. And we also used annotations, but this is sort of switch on and switch off kind of feature. Okay, all of these features go into a neural network, then it predicts a neutral effect, and there's sort of a large or a small score. In this particular case, by the way, I have to get these animations out. In this particular case, we have a score rather from 0 to 1, just the way we historically did it. We go from -100, which means a very strongly predicted to be neutral, to +100, very strongly predicted to be effect variant. Over performance, again, I said we shouldn't do that. In this particular case, we applied to a bunch of proteins. Bunch means in this particular case 4 proteins that had not been used in training, for which there's a lot of experimental data. We looked at methods, they somehow look similar. We looked at, so here's something else, accuracy for effect. So this accuracy is precision, or whatever the correct word is. I keep referring to it as accuracy. What I'm talking about is, in this particular case, where I talk about effect, you would typically call that true positives, by true positives from plus false positives. And this is true negatives divided by true negatives plus false negatives. So this is the degree, let's call it the precision for the negatives, and this is precision for the positives. Okay, this is called NPV, is that true? I can't remember what so what typically is defined here, this one is called precision, this one is called recall. Since I'm now talking about the negatives, the words completely change. And I can't remember what they are called. But they are the analogous situations, okay? And now we see that, I'm sorry, the method works very differently for buried and exposed residues. It works better for buried residues, at least for effect. For neutral, this is inverted. Initially, what we actually did is we had 3 different networks. We had 3 different networks depending on the prediction of solvent accessibility. Later, we embedded all kinds of additional features, and it turned out that to put that as an additional input unit worked better. So which of these 2 is right? Should I have 3 different networks that do it, or should I use it as an input? You cannot really tell. Try the 2 variants. The scaling through this, of course, goes through the fact that I have stronger and weaker predictions. In this particular case here, this both is predicted as effect. This is strong, this is weak. And the strength is plotted somewhere here. That is the cumulative percentage of predicted. That's cumulative accuracy or precision for the positives at least. And again, it's the neutrals, the negatives, and the positives here, the effect variance. And the reliability index, when I predict when I predict for every single, so if I take any prediction as positive where this one is larger than this, where that unit is larger than that, right? Then I get the point for the positives here. I predict 100% of all the positives. I always predict, and I get a level of precision for that of round about 77, right? That's this number here. And if I now focus on roughly 50% of all the residues here, for which the prediction is stronger, then for those the accuracy gets something like 88. Okay? So this is a tool that allows people to zoom into the better predictions, and essentially for that you have to extract the score and see whether that score fits or scales with performance. Now, in the red one here, I have a straight line. In the green one, I don't. Is that reason for concern? Or a relatively straight line. So the red is clearly more linear than the green one. Does that concern you, or does that worry you? Cannot quite expect that. And actually we have to, every single time we do machine learning, we have to verify that this correlation exists. Not only do we have to verify it just to make this point, but we also need, in a practical sense, to verify it, to give this as a tool to the user. Because now the user can say, well, if I took, in my particular protein, this fraction here, say I looked at 5% of all the residues, then I would expect that if I take that many in my particular protein, one of them is right, or something like that, right? You can make up these numbers. Now, on the next one, I'm going to show you 3 different types of points. So I'm looking at severe variance in red, neutral variance, or strongly neutral ones in green, and intermediate in blue. And I put it onto the same score. Now, what this one makes, what you immediately see, is not a very clear picture or graph, but what you clearly see is that red is on that side, green is on that side, and blue is somewhere in between. Okay? Now, what it means is, it is more difficult to predict intermediate. And in some sense, this is similar to this one. Because intermediate are the ones where the decision is not that well. But intermediate here has been, this coloring here has been done according to the experimental effect. Intermediate means less effect. Red means more effect. Green means absolutely clearly neutral. Okay? So this is no longer a binary classification. On the slide before, I trained the system to have a binary classification to neutral and effect. And now I actually sort of take out a data set that has 3 states. And I ask the third state in between, for which I never trained. Where is it going to end up? Somewhere in between. Now you can argue, because my system had not seen that. In this particular case, we argue this is not the case. And again, we collected data for that. We'll not show you. But ultimately what you see is that the strength is put into the system again through the training process. So it has learned strength. And the reason why some patterns, or the way you call that, some motives, are very different or stand out very much, is because the experimental signal for those is so strong. Okay? That's why they stick here. Very strong effect, very clear signal, very easy to predict. So the machine has worked. The machine really has learned this issue and has learned the strength of the interaction implicitly as well. Now, annotations help sometimes when they're available, but not often because they're not often available. But let's now look at a different way of looking at it. So the simplest different way is, again, what I showed you before. I have the method that's machine learning, the first generation here, that take absolutely no parameter to fit, that take no data set collection, and then I have random. That is a Q2, so it's the overall per residue accuracy. How many of the residues are correctly in the data set are correctly classified in one of these 2 states? Neutral, effect. Okay? Now, say you had developed SNAP. Would you feel good? What would you? So let's talk. I see overall. These 2 methods are very similar to each other. So this is essentially taking a blossom matrix. This uses the specific information about a family. So the green is better than the blue. Not by much, but a little bit better. But let's just take my data into 2 parts. Let's look at all the residues that are predicted correctly by both of these methods. Or let's look at these 3 methods, and let's look at everything that 2 of the 3 get right. Okay? And now say where 2 of the 3 say the same thing, not get right, say the same thing, sorry. But 2 of the 3 say the same thing, we say this is a simple pattern. This is something that anybody can do. Okay? Let's just zoom into the ones where they disagree. How well do they do for those where there's disagreement? Not 2 disagree, where they all get it the same. I'm sorry, I should have said all 3 are getting it the same. So here is the subset of the ones for which there is some disagreement between the 3 methods. And the question again becomes, for that subset, how well do they do? Now you see that these 2 here fall below random. Your immediate reaction may be, okay, why not flip these methods into a coin? You cannot think that way, right? Because we threw away everything for which they were made, so to speak, for which they worked. And so now for the subset, actually, it's just like that. If I took the entire set, they do way better than random. So turning them into a coin is really doing much worse, right? But for this subset here, they do actually worse than random. And SNAP does worse than it did before. It sort of reached here. But it does way, way, way better than anything else. And ultimately that sort of is the reason to argue why this machine learning makes a big difference. Because for the ones that are not the simple patterns, the machine learning is the only thing that really gets them. Okay? And again, it's specific alignment information does way better than just the blossom matrix, but this is way better even. So when Max Hecht sort of went into the next round doing SNAP 2, he tried all kinds of things. He put in disease genes as opposed to the ones we had before. He put in all kinds of more additional input units, tried to increase the neutrals, the enzyme story, looked at additional independent data sets, looked at more input features such as coevolution, more physical chemical features. You see that the length of things that made a difference has increased here, and he put special versions in that actually don't use alignments at all, or that don't use disease data. Now this is relevant because now you can say, well, if I for one particular variant predict that there's an effect, and I do this too here, then this means I predict an effect because of something that happens to the protein that has nothing to do with the alignment. It's something in the biophysical features. Okay? If you don't use disease data, that means that what you have is a molecular effect as opposed to something that happens on the organism. And again, gets sort of slightly higher scores. And he does again this, where one of them does better than the other. Now the numbers are no longer that good. PolyPhen, in fact, has meanwhile improved too. But it's still true that the method here does, for the ones that are not trivial, way better than anything else. And I believe this is where I will leave it for today. I will not leave it for today. Are there questions? Because if not, then I'm going to go into the next lecture. That I will continue on Thursday, and I go into it today because on Thursday it may be a little bit too long for Thursday. And that is more about the question: once I have these prediction results now, what can I do with them? So let's first look at disease data. So one way of looking at disease data, one set of disease data are variants that are referred to as OMIM variants, online mammalian inheritance in man as a database, and the database is populated by about a little bit over 1,000 variants, SAVs, single amino acid variants, that have an effect on the carrier. Some of those, in fact, are diseases. The sickle cell anemia is one of these 1,000. Okay? And many of those are, in fact, bad diseases. Now, one way of looking at it, I lost my story here. I'm sorry, I cannot predict. Okay, I lost my story. So I will get back to that story before. Ultimately, I cannot remember exactly what I'm drawing here. That is, I'm losing my story. The result at the end of the day is that when I look at OMIM variants and I predict OMIM variants, then I see that the prediction strength in terms of the reliability index, for instance, is higher than the one that I had for my training set. So I have, for the first time in machine learning, we had a data set of thousands where, in fact, my training performance was bested, or I did better than my cross-validation. Okay? Usually, when you get a new data set, you do slightly worse. Usually, whatever you do in terms of cross-validation, you do your best, it will never be good enough. That is our experience, at least. You may again get close to being good enough, but you will always, with a new data set, do a little bit worse. That's the best scenario, that you do a little bit worse than you estimate. And now we suddenly have a data set where we do much, 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 much better than we expected. And then the question, of course, becomes why? And the answer to that is, ultimately, again, I'm sorry for the confusion that I have in my head at the moment on explaining the data, but ultimately the precision for this OMIM variance, where I have 1,000 variants in human that never seen before, and the precision for getting them is way better than any data set that I use for cross-validation. Why could that be the case?
Maybe the data is similar because it has strong signal, because it's a disease.
Exactly. So ultimately, we have the story that I showed you with a extreme experimental effect, extreme experimental neutral, and the intermediate, the blue, where I said that if it has a strong experimental effect, it tends to be predicted more on the right side, more on the side of effect, more on the side of where in fact we know that the predictions are more reliable or in fact perform better. And this is what I have. I have a data set that is experimentally so clear, so clean, this is a dramatic effect. Okay. Now, I can use this data, or you can use the same method and ask, so OMIM are variants in human, single amino acid variants that cause disease. And I'm going to get a certain value in terms of what fraction of those I predict to have an effect. Okay? Now if I did the same story for animals, so for single variants in animals, so I'm going to show you on the next slide 2 bars. One bar will be for man or woman, OMIM. Let me sidestep the issue of man and woman, OMIM. And one will be for OMIA in animals. So which one will have a stronger effect? Will they be the same? Will they differ? So we're talking about variants. Every single one of those is known to cause a disease.
What is OMIA again?
Animals. So it's the same. This is human, it's Homo sapiens, and this is animals. It's a bunch of animals.
I think on the animal we would have better because we have more data.
No, actually, it turns out not to be the case at all. So a lot of the experimental data in molecular biology is human-centric, and a lot of the animal data is not that easily parsable and available. So there is some. It is true that there is some more data, but the question is not really what is the number of data points here. The question really is what's the effect. So are the variants that cause disease in animals, are they predicted to have more effect or less effect than the ones in human? Again, none of those had ever been used for setting up the device. Okay, this is an additional data set that is very clean in terms of cross-validation because I never used the data. Will animal be the same as human? So the answer is it is not, actually, and it's a tiny difference, but since we have thousands of points here, the difference does matter. Well, here we have only 100, but still, we argue that the difference matters, and ultimately the argument why this one is higher than this one is because this contains things such as change of hair color. Okay? It's not really a disease, but if you are a happy blonde and then you suddenly turn into brown, maybe you're totally unhappy and you feel this is the worst disease that could have happened in your life. For the animal, the dogs will typically not complain about the change in hair color. So all the variants listed here are really dramatic things, right? You see something that everybody immediately, and that ultimately is exactly the difference we see. Then we can actually go and do the, I should have taken the other one. So we can ask, if I put the OMIM into mouse, this is what molecular biologists call a mouse model. Or as opposed to the ones where I have the same variant, I put it into the mouse. So what I do now is I have a mouse sequence here. And in this mouse sequence, I vary this E to V. And I vary E to V only if mouse has an E. Okay? That's the right one. In the left one, in the human here, I essentially change the E to V no matter what. Okay? I just sort of humanize the mouse sequence. And this has a stronger effect And this ultimately is exactly what we do with animal systems. So we sort of import a human variant into a different sequence, into a different protein. Yes, it's similar. Mouse looks in terms of, mouse looks much more similar to us than a fly. Okay, it's a mammal and so forth and so on, and it's much easier to do experiments with mice than with people, and certainly it's also easier than pigs or something like that. So the more similar the animal is to us, the more difficult it is to do experiments with them, and that leads to a situation that mice are many do a lot of experiments now in fly. Because again, it's easier to mistreat the fly than mice. But anyway, what you see is that you're not getting the same effect. So if these two here would have exactly the same effect, then it wouldn't matter whether you use mouse or human, right? But you see there's a big difference, and the prediction method can see that big difference, okay? So that is surprising. So what you see essentially is a method that can not only predict the effect of variance, it can make statements about how good is it to use mouse as a model system for human. Be careful, because you're not going to see the same thing. Not only because of some molecular issues, but simply on the mere level of the protein. You're messing up something. Okay? It's not only because the mouse has more immune system, different immune system, or can handle things different. On a much smaller molecular level, things already go wrong because there's a differential effect, because the sequence is different. Let's not get into that. Now, another way of doing this, and I believe this one is what I'm going to go into on Thursday.